Okay, thank you, everyone. We're glad that you're joining us. For those of you that are keeping track of this, we're kind of in our second round of rotating through the various NIAMS funded CCCRs. And so we're leading off, and we have a couple more coming up for the next two scheduled, and we're planning to do this roughly on a quarterly basis. So you can be looking for those invitations as well. So we're glad you're here. Like I said, I want to introduce our speaker. Nikki Freeman is a postdoctoral research associate in the Vascular Division of the Department of Surgery in the UNC School of Medicine. She received her PhD in biostatistics at UNC under the direction of Dr. Michael Kosterok. Her current work and interests are at the intersection of precision medicine and translational science. Her presentation today is titled Individualized Treatment Rule Characterization via Surrogate for the Value Function. So thank you, Nikki.
Great. Thanks, Todd, for the really kind introduction, and to Abby for being an amazing organizer. It is delightful to be with all of you today. Just one note, I am going to remain in speaker mode right now. Can you see the little bubbles next to me, or just want to make sure everyone can see?
Yep, we can see them. Yep.
Okay, cool. So I'm going to stay in speaker mode. So I know sometimes people share screen, but I'm going to stay this way just so that we get a little bit more FaceTime and it's not just slides. But because of that, I have to do this weird kabuki with all of my screens. And so I can't actually see what's going on in the Zoom. Anyhow, the title of today's talk is ITR Characterization via Value Function Surrogate. It's great to be with all of you. Okay, so I like to start out with the origin story of how all of this comes along. And I work very closely, kind of one slice of my work with Kate McGinnigal, who is a vascular surgeon. And full disclosure, most of my applied work is with vascular surgery and diabetes. So I'm not, I have no expertise and very little skills in rheumatology, although I'm going to try to make this as applicable as possible. But I work with Kate McGinnigal and then Michael Kosterok, who I think many of you might know. I know he's a part of the group at UNC, just former chair of Department of Biostatistics at UNC, and, really one of the pioneering leaders in precision medicine. And then there's me. And my husband's upset because I put that I do translational science. And he says, well, you do precision medicine too. And I said, yes, but it, makes the figure much nicer if I have these kind of three branches, which are the three ideas that we're going to talk about today. So all of this to say that these are kind of the three ideas that are all going to come together in this presentation. All right. So the first idea, just as a little amuse-bouche or a little preview to get us going, is the idea of precision medicine. And we're going to talk about this more in a little bit. So I just want to clarify up front that when I say precision medicine, I'm thinking about statistical precision medicine, using data to learn how to match treatments to individuals based on observable characteristics, which is different than other ways that the word precision medicine is used, like in precision public health or in like more mechanistic understandings of disease and treatments, where you're really thinking about what is the mechanistic way in which genes and other kinds of biologics work together. So that's what we're going to be talking about. So when I say precision medicine, I'm thinking kind of in this statistical framework. The other idea that I really try to center my work on, and what a lot of this work that I'm going to present to you today is about translational precision medicine. And I'm just going to read at you this definition: that translation is the process of turning observations in the laboratory, clinic, and community into interventions that improve the health of individuals and the public, ranging from therapeutics and diagnostics to medical procedures and behavioral medicine. And really, when I'm thinking about this, I'm thinking about how do we bridge the gap between precision medicine analytics and all this very fancy stuff that people are doing. So like machine learning and deep learning and, you know, all the different kinds of learnings, Q learnings, reinforcement learning, all that stuff. How do we go from the evidence that's generated there to clinical and public health practice? And so if you've, you know, spent a lot of time like looking at like the resources that come out of like NCATs and, you know, think about translational science, a lot of that is really about how do you build the teams for this kind of work, and who should be on those teams, and how do we really think about moving evidence forward and making it actionable. And of course, I'm biased as a biostatistician, thinking that, you know, biostatisticians are absolutely critical for this. And I think in precision medicine, especially, there's these huge opportunities for statisticians to really kind of help bridge that gap and start moving into new questions that we could be asking as we're developing precision medicine evidence for different disease contexts. And then finally, I just want to be a little bit more specific about, you know, why do we care? What am I actually going to talk about today? And so I'm going to try to kind of ground this in, I think, work that many of you might be already familiar with. So the first thing is, you might know Phoebe. Phoebe was a biostat PhD student here. I think she graduated in 2020. She was one of Michael Kosrock's students. And while she was here, she was the first author of this paper, Precision Medicine Approach to Develop and Internally Validate Optimal Exercise and Weight Loss Treatments for Overweight and Obese Adults with Knee Osteoarthritis, data from a single center randomized trial. And if you've read that paper, it is really just like the playbook on how to do really thoughtful, state-of-the-art precision medicine, DTR learning, optimal dynamic treatment regime kind of work. And it's really like this beautiful recipe of like how to do this. And you might recognize some of the other names like that are in the author list, because it was really just like a lovely work of how to do precision medicine and bringing together a lot of different people. And one of the things that came out of that paper was this treatment rule, which we're going to talk about what treatment rules are in just a second, just make sure we're all on the same page. But basically I said, if you want to maximize weight loss, then there are these two conditions. And it's like if your baseline weight is above 109.35 kilograms and your waist circumference is above, you know, a certain number of centimeters, or, you know, some other combination of weight and waist circumference and prior MI, then the optimal recommendation to maximize weight loss would be to recommend both diet and exercise. Otherwise, just recommend diet alone. Okay, so that's an example of a treatment rule where it's individualized because it's based on these observable things that we can see, you know, when someone comes into the clinic or comes into for routine care, weight, waist circumference, and, you know, history of prior MI. Someone else you might know as well is Xian Kim. She also worked with the group here at UNC. She graduated with her PhD this summer. And she's been working on tree-based methods for developing highly interpretable treatment rules. And so you can see here's an example from her work where, you know, it's this decision tree and you're looking at BMI and other kind of factors for making optimal recommendations. And I think both of these works are beautiful examples of what we have been trying to do in precision medicine, which is to develop these rules on how to optimally treat individuals based on their observable characteristics. And so my question, and the question that came out of my work with Kate McGinnigal, the vascular surgeon, is she would ask me things like, well, what if this isn't really quite feasible for me to follow this rule? And I kind of acted not counter to the rule, but just adjacent to it. What would that mean? Would we lose all the benefits of precision medicine? Kind of in a more kind of pointy-headed statistical way of thinking about this, you might think about, well, these are optimal rules, but that doesn't mean these are the only optimal rules, or that there might be multiple rules that give us the same clinically meaningful effect, but maybe some of them are less resource intensive than others. So like in Xian, you can see like some of the recommendations are for like PT, which is highly resource intensive because, you know, you need to have a physical therapist there, you know, actually delivering the intervention. So that is kind of where what I'm going to talk about today is thinking about how can we characterize these rules more generally. And the idea is, like, can we start bridging that gap between, here are these wonderful, excellent, optimal dynamic treatment regimes. How can we contextualize and help to understand them when we start actually start thinking about the contextual factors that go into clinical decision making? Okay, so that's the themes of today's talk. So precision medicine, of course, we're thinking about translation and then trying to motivate this by really kind of where the field is. I do want to just say a little bit and be a little bit more specific by what I mean when I say precision medicine. So we've all heard right treatment, right person, right time. But formally, we kind of need to have some notation, and I'm going to try to be as notation light as possible today. So a lot of this I'm just going to say with words. So we're going to imagine that we have a single stage problem, and we're going to denote that with a K. And we're going to assume there's just one decision point. And we're going to assume that there are two treatments, the 0 and the 1, and we're going to denote that with an A. We have observable patient features, which are these denoted by H for history. And there's some outcome Y of interest. I've assumed that it's a real outcome. It doesn't necessarily need to be that. And just for ease today, we're just going to assume big Y's are better. But of course, if you wanted small Y's, you could always replace every time I say maximize with the word minimize. Okay. So then this next thing that you see, this dynamic treatment regime or individualized treatment rule, different literatures kind of give different names to this. So some people tend to call a single stage rule an individualized treatment rule, and if it's multi-stage, dynamic. Some people say it's all dynamic. I am going back and forth, and I'm really sorry about that. So just full disclosure that I say both interchangeably. But the whole point of it is that you have this function D, and it takes in patient observable characteristics, and it pumps out a recommended treatment. So take in history, get out a possible treatment at that decision point. And there are kind of two things that we do a lot in statistical precision medicine. And the first one is we do something called evaluation. We say, how good is a particular rule? And the way we do that is by estimating or calculating the marginal value of a DTR. And so I'm denoting that with this script V of D. And it's just the expected value of the outcome we would observe in the population if everyone were treated according to treatment rule D. Okay, so what if we all decided to follow this rule? How well would we do on average in the population? The other thing, so that's evaluation. So that's really important. The other thing that we're interested in is finding optimal dynamic treatment regimes. Can we find this rule D opt so that it is the best rule of all the rules that we're considering? So if we were to follow this rule in the population, then everyone would be, on average better off than if we had followed any other rule. So evaluation, optimization, kind of the two very common tasks in statistical precision medicine. And of course, I have said all this in terms of potential outcomes. What would have happened if everyone had done, blah. And of course, we want to estimate these values and these optimal rules from the data. And so how do we get from potential outcomes to, identifying that from the data? We're going to need some assumptions from causal inference. We're going to need causal consistency and SUPA, positivity, no unmeasured confounders. And I'm just going to hand wave those. Those are like the standard assumptions. And we're going to assume that everything is okay for now. Okay, so that is precision medicine. So let's get to the actual meat of the presentation, which is, this idea of characterization. So I talked about two main ideas that we do in statistical precision medicine, which is evaluation and optimization. And, I'm going to argue that there's other things we should be doing. And one of those that I think we should be doing is characterization. Okay, so the goal here is to characterize, and I'll tell you what I mean by that in a second, and find optimal tailored treatment roles. And this work was inspired or, kind of motivated by work from chronic limb-threatening ischemia. So I have on here, non-healing, slow-healing wounds for patients with CLTI, but really this could apply to any kind of treatment rule. And our strategy is we're going to do characterization via a Gaussian process surrogate for the value function. All right. So what does that mean? Well, remember we were just talking about what is precision medicine, and we formalized this. And we're going to imagine that we have some particular treatment rule D, and the value of it is just expected value we would observe if everyone were treated according to that rule in the population. Okay, so that first equality is exactly what we just talked about. That's the definition that I gave you. And then the second equality, you see, I'm no longer writing this in terms of potential outcomes. I've identified it. So now we have, actual data in there. And that's just from all the hand waving that we did from the causal assumptions. So I can estimate the value for a particular treatment rule if I can calculate this iterated expectation. Now, the next line here is I'm telling you a little bit about the class of treatment rules that we're going to be thinking about. And this is super ugly, so I'm just going to tell you with words that we're just imagining that we're considering treatment rules that have a finite parameter, and that it's bounded and that they're real valued. So, this could be very simple things like kind of like Phoebe's rule, where if your weight is less than 109 kilograms, like that's one parameter. If your waist circumference is within another parameter, that's the second parameter. So a finite number of parameters, and they're bounded, right? So like they're not going to wander off to infinity or negative infinity. And so in the line that has the two in parentheses next to it, really all I've done here is I've just replaced everything that we believed in line 1 and just really kind of hammered home the point that we're thinking about decision rules that are functions of theta. And because it's, finite theta, we can really then think of the value as just the composition, right? So the value is, is a function of the treatment rule. The treatment rule is a function of theta. So if I really wanted to optimize the value rule and find an optimal DTR, what I really need to do is just find the theta that optimizes the value. And that's what that last line is saying, is that, really when you've assumed that your rule is finite parameters like this and bounded, you really just need to optimize the value function as a function of theta, because each of those thetas index a decision rule. So this is really cool because we have kind of a simple problem, right? Like I can think of the value function over the treatment rule space as a value of the treatment rule parameters. And this seems really straightforward and nothing seems too scary or crazy. There's a little bit of bad news though, is I haven't really told you about what this V looks like. How do we actually do that estimation of the V? And we know that we can estimate it because I just told you we can do evaluations. So we know that there are ways, but it may not be, friendly, right? It may not be differentiable. It may not be fun to work with. And, of course, there's always just brute force, right? We could just grid search over this parameter space. But that seems, really arduous and hard and not a good time for someone's graduate student. Okay. So what we're going to do instead is we're going to use the tools that we have for point evaluating the value function. And you've probably heard of some of these. So, inverse probability weighting, augmented inverse probability weighting, G computation. These are all strategies for evaluating the value of an individualized treatment rule. And so what we're going to do is we're going to sample some thetas, okay? And remember, sampling some thetas is the same as sampling some decision rules, because the decision rules are completely parameterized by these thetas. And then we're going to use the tools that we already have and we know and we love, IPW, AIPWE, G computation, and we're going to evaluate the value at those thetas. And what we're going to do is we're going to assume that there's a Gaussian process prior on the value function. And by Gaussian process prior, we just mean that the value function is some smooth-ish function of theta. It's just some kind of function. And then we're going to use our beliefs. We're going to update our beliefs using those thetas that we sampled and the V's, the values of those particular rules that we calculated using AIPWE, IPWG computation, whatever's your favorite flavor. All right, so I'm going to say that one more time and be slightly more specific, but like not too specific. So we are going to start out with some thetas. Remember, sampling theta is the same as just sampling over different decision rules. I'm going to pick my favorite value function estimator to get estimates of those values. I'm going to assume that the value function is some function and that the prior on it is a Gaussian process with some kernel. And I've in the slide here, I've centered it at 0. And that's, just because that's kind of like the standard way you would do the Gaussian process. And then I'm going to use this theta 1 through B and these associated V hats that I've calculated to update my posterior about what I think the value function looks like. So we've fitted this Gaussian process. Okay, so I have all of these, I've sampled some spots, some different treatment rules, I've estimated their value. I've updated what I believe the value function looks like. What can I do with my fitted Gaussian process regression? And we're going to be able to do 2 things. The first thing is optimization. So we're going to be able to use tools from Bayesian optimization to find an optimal. And I've written here the expectation or the expected improvement algorithm on how to do this. And this is really not essential. This is a very well-used, common strategy in Bayesian optimization, but I'll just give you the basic idea is that you have your Gaussian process, which is your estimate of the entire surface of the value function over the treatment rule parameter space. And then you're going to use this function here, EI, the expected improvement, to try to find where you think the optimum might be. And it's this trade-off between exploitation and exploration. And this EI is very simple to calculate and optimize over. And so instead of having to optimize our value function, which might be weird or we don't know what it looks like, we can work with this EI, which is also called an acquisition function, and tells us what point should we be acquiring next to really try to learn where those optima lie. And so the expected improvement gives us a new point, a new theta. It says you should go here to get the most improvement. And then we'll calculate its V hat because we have tools for that, IPW, AIPWE, G computation, whatever. And then we'll update our Gaussian process. And we're going to keep doing this until we get, we've basically kind of converged at an optima, or we say our sampling budget is met. I'm tired of this. I'm done. I'm, going to go get a snack and go take a nap. Okay, so that's optimization, really just kind of off the shelf expected improvement. The second thing that I think is possibly more interesting or useful is this idea of characterization. And this is using the posterior predictive distribution from this Gaussian process regression to characterize the entire treatment rule parameter space and the value of those treatment rules. And I think this is easier just for me to show it to you. So I'll show you some simulations and then I'll talk to you about how we did this in vascular surgery for CLTI and wounds. Okay. So does it work? And the answer is yes. I would not show you simulations unless they worked. So let me orientate you to what I'm showing you. I used decision rules that have 2 parameters, beta naught and beta 1. So the beta naughts are on the horizontal axes, and then the vertical axis are the beta 1s. And these are the same as the betas in the last set of slides, but because, brains are really funny things. For some reason, I switched over to beta when I wrote the simulation. The W's at the top, these are just different noise levels. So these are just 3 different noise levels at which I ran the simulation. And then the color corresponds to the value of the treatment rules that associate with those 2 beta naughts and the beta 1. Okay, so if you were to pick a beta naught and a beta 1 and go where they meet, those 2 define a treatment rule, and then the color corresponds to their value. So we can see that these lighter colored regions are more optimal than like the darker color region. So if we were going to try to find an optimal treatment rule, we would want to find those that put us, say, pick the ones that have parameters in these lighter regions. Okay, so that's what this is. So these are the true values. And then in the simulation, what we do is we use AIPWE, G computation, IPW, and stabilized IPW to estimate treatment rules. And then we estimate the values of those rules. And then, we find those treatment rules and we plot them on top of the picture I just showed you. Okay, so we really want rules that are picking optimal beta naught and beta 1. And you can see that for the most part, everybody's ending up in those light colored areas, which is exactly what we want. There's one exception, which is G computation. And that's because we super misspecified the models for G computation, and it failed horribly. And that's just one, and I put this in here, not to tell you like, wow, model misspecification bad, but really just that, this really does depend on having a good value function estimator. And so, if you have this kind of problem, this can go really awry. What I think is interesting, if you recall, the truth, which is on this slide, there are multiple optima, right? So like if you look at that right-hand panel, there are these 3 regions that are yellow, and maybe whatever the context might be, 0.2, 0.3, this is all like really good, maybe clinically equivalent, and we'd be happy to end up with a rule from any one of those. And across different simulation runs, you get different rules that are ending up in different places. But if we had only done this once, and that's what I did here, this is just if I did it once, the rule that I learned is where that red star is. And I would have missed out on the richness of knowing that there were these other optimal regions that I could be exploring. And so what I'm showing you here is the optimal rule that I learned through the Bayesian optimization, the expected improvement procedure, is the parameters that correspond to where the red star is. So if you start at the red star and go straight down and straight out, those are your beta naughts and beta 1s. That would be your treatment rule. But the color on this plot, this isn't the truth. This is me just plotting out the posterior predictive distribution from the Gaussian process regression. And so what I'm learning by thinking about this, I not only have an optimal rule, but I can also see rules that have similar values that would lead to similar outcomes that are nearby, some that are a little bit farther in kind of this other space over here. And if I just focused on the optimization task, I would have missed that altogether. So, my thought and my recommendation is, there's more for us to do than just optimization, that this kind of characterization work is important, because maybe some of these rules are more feasible, maybe they're less resource intensive, maybe they're more acceptable depending on your particular patient population or provider preference, whatever it might be, that, instead of me just giving you one optimal rule, I can give you this kind of whole map of what all these rules might look like very quickly, very efficiently. And then we can explore, there's this other yellow region. Maybe we should look at rules over there. Like, what does that mean? And in a real context, is this better? Is this worse? Or, how can we use that kind of information? And so that's what I mean by characterization. As opposed to just handing over a rule, we can kind of look at all the rules at once and think about what those values mean. All right, so I'm gonna switch over from made up things and made up simulations to a decision rule. And I don't have one that is rheumatology focused, because I don't have any data for that. So I'm gonna talk about CLTI, but it's pretty straightforward. Chronic limb-threatening ischemia is the most severe form of peripheral artery disease, which is narrowing of the arteries in usually in the lower extremity. And because of the reduced blood flow in the limb, people often get wounds, and then those wounds don't heal. And so then there's this question of, can individuals heal their wounds? We assume that they're not, like a grade 3, like these terrible infections and whatever, but these wounds are not healing. Is the best course of action to try to do non-surgical wound care, or to revascularize these individuals surgically. So either bypass or through some kind of endovascular procedure. And so that's what we're looking at. And one thing that's interesting about this wound care versus revascularization type procedure is, and this is what Kate and I were thinking about and working on when we were working on this analysis, was for wound care, the way it works for these patients is they have to show up at the wound care clinic. And so there is this like compliance kind of contextual aspect of, can you actually adhere to a wound care schedule? And some of that might be just, individual motivation, but some of that's also just, there are structural barriers, lack of transportation, scheduling jobs, childcare, whatever it might be that would prevent that, as opposed to revascularization, where you show up for surgery and then hopefully your surgeon does the thing. So the data that we had for this, it's a single institution cohort of EHR data that we linked to administrative scheduling data from one of the wound management clinics in the UNC system. And the way we looked at compliance, because this is really what we were interested in, was looking at compliance and how it affects, how you should use compliance and past compliance to make treatment recommendations. So if your patient hasn't been coming to the wound management clinic, how should that weigh into your decision about whether to continue with the non-surgical pathway versus surgical treatment of the reduced blood flow in the arteries? So we measured compliance as completion of scheduled wound management visits, and we assumed perfect compliance or perfect adherence for revascularization. And the way that this works is UNC is a limb salvage center. So everyone, unless it's like an emergent limb, gets wound management and they receive wound management initially. And then, after a while, people kind of see like, oh, maybe this isn't really working. Maybe we should really start thinking about a revascularization. So after a month of a trial of wound care, the clinicians decide whether to continue with wound care to revascularize. And then our outcome of interest is the wound healed? Is the patient alive, and is the limb intact 6 months after the initialization of wound management? So let me show you the type of rules that we were considering. And so this looks very complicated. It's actually just, I wrote this very poorly. We're looking at rules that revascularize if the baseline compliance during that trial of wound care is less than θ1, or if the initial wound size is bigger than θ2. Okay, so that's this very simple rule, things that one could very easily figure out. There's no special kind of test or measurements that need to be taken. Very straightforward, simple rule. So it's a 1 if your compliance was less than some threshold or your initial wound size was bigger. It's a 0, so continue with wound care otherwise. And I'm just going to show you the results of this. So at the very bottom, I've written down what is the estimated optimal rule, which is if you went to less than about 25% of your wound care visits, or if your wound was bigger than 89.8, I think it's like centimeters squared, then after your trial of wound care, if you're not making good progress, you should be revascularized. Otherwise, continue with wound management therapy. And so in the picture, this looks a lot like those simulation ones I showed you a second ago. We have baseline compliance on the bottom and initial wound size on the vertical axis. The white star is the optimal treatment rule. So what's written there at the bottom, that corresponds to those parameters. You can see it's in this, and so that's what that is the white star. The bottom, the color corresponds to the surface of the Gaussian process regression that we did. So this is giving us the estimated value across all of the different, baseline compliances and initial wound side combinations. And what you see is that there are actually, a lot of rules, a lot of ways that individuals, patients and their providers could act and make decisions that would keep us within this dark red and bigger is better in our world kind of region. And so there is some wiggle room per se. The other thing is it's interesting just to see how quickly kind of there at the bottom, value starts to drop off when you change in the wound size direction, or how slowly it's changing in the compliance direction. So that's kind of interesting. I think Kate thought that was interesting too. And then finally, all the little black dots, those are all the places that we evaluated as part of getting this surface. And so really, I only evaluated black dot many amounts of times plus the white dot, and then was able to get this entire surface that characterized, all the treatment rules in this class of rules with this very simple setup of looking at compliance and initial wound size. So I think it's a nice result, and I think it also just shows that yes, we have this optimal DTR, but if you're a little bit off from it, or what does that mean? How does that happen? And helps us kind of think about how could we use these when context really matters. All right. So that's it for the kind of meat of the presentation. I just want to thank you all again for having me. It's really great to meet different groups. I have 3 really great co-authors that helped with this work. Sydney Browder is a UNC epidemiology PhD student. She did a lot of the data cleaning, which is fantastic. Administrative data is really, really gross, and she was a really good sport and did a great job with that. Kate McGinnigal and Michael Kosterok are my 2 collaborators. This was some funded work with funding from National Institute on Drug Abuse and from NCATS. You're welcome to email me if you have questions or thoughts. And then I just put on here the link to the Precision Health and AI Research Lab, which is the lab that Michael Kosterok and I lead. So yeah, great. I think that's that, and I'm happy to discuss or take questions or whatever.
Okay, great. Thank you so much, Nikki. We do have some time for questions. And so we can questions if you want to unmute yourselves, or if you want to type something in the chat. And I'll lead off. So this is impressive work, and obviously there's a lot that goes into this. There were a lot of formulas and computing and so on. Could you just talk a little bit about what is involved in terms of some of the extent of programming or coding that you need to do, as well as maybe some of the software that you're using for these projects?
Yeah. Yes. So for the heart of it is the Gaussian process, right? And getting to that Bayesian optimization piece. And all of that was done in Python using off-the-shelf kind of Bayesian optimization packages. And so I think that's one really nice thing, is I did not code up my own expected improvement. And then I was able just to, out of some of the metadata, just kind of shake out the points I needed for the posterior predictive distribution. And so it's really not about like heavy programming. Like the programs are actually really quite short. It was just being able to kind of dig in there and find what I needed.
Great.
And then I should say, I feel more comfortable plotting in R. So all the plots are in R after I kind of shook everything out of Python.
Gotcha. Lubov has a question.
Yeah, I just it's a general question. So like, imagine if you have a dynamic treatment regime that can change, and time varying covariates that can be affected by these treatments every time. So what is the advantage of using a G formula instead of inverse probability weighting?
So there's a trade-off there, right? So inverse probability weighting, you're only using the people that got that particular. So if we're doing evaluating the value of a rule, with IPTW, you're only using the people that actually followed that rule.
Okay.
With G computation, you're doing some modeling, right? And so then you can use everything. Yeah. IPW is unbiased, but it has high variance, which is why Maybe sometimes it's not the best. But as you saw, like in my simulations, G computation, if you get the models wrong, you're in trouble and really bad things can happen.
Okay.
So I don't think I'm really answering your question about what to do with the time variant covariates, other than that, IPW versus G computation, there are pros and cons to both.
A lot of papers, yeah.
Yeah.
And another question is like, why is it possible to make an assumption that the process is Gaussian, but what it's not, if it's not?
Oh, yeah, thank you. That I should have clarified that. So I'm not assuming that anything is Gaussian distributed. I'm assuming that the value function itself, so the outcome we would observe if everyone were following according to a particular rule, that function has a Gaussian process prior. A Gaussian process prior is a prior over functions. And so basically how I pick the kernel for that is telling me how squiggly or not squiggly I think those functions look like over the value function itself. But I'm not saying like, our outcome is normally distributed. That's not the assumption.
Yeah, so this is like more Bayesian methods. So you have priors. Okay.
Yes. So basically it says, if you were to just draw functions from a particular Gaussian process, you would get, all sorts of functions going every which way. And then you update it and it says, okay, maybe it's not so squiggly over here. I have a little bit more information. Maybe I have less information here. So maybe there's, more variation, but, you start trying to narrow in on what that function shape looks like. That's what the Gaussian process prior is doing.
Okay. Thank you.
Other questions? Nikki, I'll note that Amanda Nelson put something in the chat complimenting your visualizations.
Oh, thanks, Amanda. Yeah, I started out kind of when I was trying to figure out how do I get into public health research, working in like meta evaluation and meta analysis, and I spent a lot of time building like dashboards for those kinds of results. And so I think I feel like pictures can just tell you so much more than the math on a slide. So yeah, thanks so much for that.
I had another question, and again, this is less technical, but I'm just curious to hear what your experience has been in terms of working with, say, clinical collaborators. Obviously, you showed a very neat and tidy sort of presentation in terms of start to finish, and a lot of times these things don't quite go that way, or they're very non-linear, you might say. Can you maybe talk a little bit about what those interactions look like in terms of being able to get to the point of figuring out, what are the features that should be included, and sort of making sure that you're on the same page and speaking the same language as, someone who's not, a biostatistician, and also for you to be able to understand what they're communicating?
Oh, Todd. Such a good question. I will say I've had a couple of really great partnerships, and where me and the clinician, me and the researcher really feel like a team, and just that kind of really just mutual respect for our skill sets, and have spent, and a lot of it is just time together, time talking, time really understanding what their deep question is, not just coming to the table with, well, here are the methods I have, let's shoehorn your questions into those methods. And so I think that's really great. On the other hand, I've had situations where these are collaborations that just didn't really pan out. We spent 6 months together, and I didn't really feel like the good vibes. I don't think they felt good vibes, and it, nothing bad happened. I think we just said, this isn't really working out. I wish you the absolute best in your work. And so I think there's so much to this work that is just beyond the technical, beyond the science. It's really just like finding that chemistry of having a common goal, like you said, developing a common language. So like when I started working with Kate McGinnigal, like I read a vascular textbook to try to understand like what are all these words and these different measures and, you know, things that she was telling me about. I think it just takes time. And then in, like the process of research, I mean, I think there's a, I don't know if you watch Succession, there's this line about the endless middle. I mean, you spend so much time in the endless middle of trying to figure out all these different things. But I think it's really valuable. You know, at the end of the day, you can really say like, we really thought hard about this, and this is, you know, the best science that we can put forward given, you know, what we knew at this moment. So it's hard work, but I think if you're working with great people, it makes it, you know, enjoyable work too.
Great, thank you.
No.
Any other last questions? All right, well, we want to thank you again, Nikki, for taking the time to meet with us today and share this work. We are going to launch a brief poll. It won't take very long, but please take a moment before you sign off completely to address that. And again, thank you for being here, Nikki. Thank you. Everybody have a great day.